Howdy and welcome to Gen Edge Live, our Zoomcast show that gets life sciences down to business. I'm Alex Philippidis, Gen's Senior Business Editor.
And I'm Kevin Davis, Editor-at-Large with Gen, the Executive Editor of the Crispr Journal and the author of Editing Humanity, the Crispr Revolution and the New Era of Genome Editing.
Gen stands for Genetic Engineering and Biotechnology News. It's the world's first publication devoted exclusively to biotech and the life sciences. We're celebrating our 40th anniversary this year. One way we're celebrating is by delving deeper into the business of biotechnology. On today's show, we're joined by three analysts who are authorities in the world of genome editing and allied technologies. Michelle Gilson from Canaccord Genuity, also Arlinda Lee from Canaccord Genuity, and Geulah Livshits from Chardan. Hello to all three of you. We'll be talking to Michelle and Arlinda and Geulah in just a moment.
Gen Edge Live is a production of Gen Edge, the premium subscription service for exclusive news and insights of critical interest to biotech industry executives and life science professionals. Gen Edge goes beyond the headlines and cuts through the noise to give early insight into how, when, and why the latest biotech developments will impact future outcomes and investments to keep you ahead of the competition and on the bleeding edge of biotechnology. Gen Edge comes with a free 30-day trial and a growing archive of articles well worth your attention. You can find Gen Edge at Gen's website, genedgenews.com.
On today's show, we want to explore some of the latest milestones in genome editing. When it comes to genome editing, there's never a dull moment, it seems. The CRISPR community is buzzing with the recent milestone published by Intellia reporting the first clinical data on in vivo CRISPR genome editing in humans. Those results, published 2 weeks ago in the New England Journal of Medicine, appear to support the broad application of in vivo genome editing to treat a host of other rare and perhaps common diseases.
And today we also saw the announcement of the launch of Prime Medicine, the latest spin-out from the lab of Harvard and Broad chemistry professor David Liu, which raised more than $300 million.
To discuss these and a lot of other exciting progress in the field, we welcome Michelle Gilson, a managing director with Canaccord Genuity focused on biotechnology. Michelle joined Canaccord Genuity in 2018, having had previous experience in healthcare equity research teams at Jefferies, Goldman Sachs, and Oppenheimer. Also joining us is Michelle's colleague, Arlinda Lee from Canaccord Genuity, and Dr. Geulah Livshits. Oh, I'm sorry.
I'll take that one. Also joining us, Dr. Geulah Livshits, a senior research analyst at Chardan, covering biotech companies with a focus on gene editing and oncology. Geulah joined Chardan in the spring of 2018, having worked as a postdoctoral fellow with Scott Lowe at Memorial Sloan Kettering Cancer Center, where she developed CRISPR and RNAi approaches to study pancreatic cancer in vivo and in organoid models. You're welcome to send us your questions for the panel through the Q&A feature on Zoom, which you're all very familiar with by now. It's at the bottom of your screen, and we'll take as many of those as we can during the course of the next hour.
Mm-hmm. Michelle and Arlinda and Geulah, welcome to Gen Edge Live.
Great to be here.
Thanks.
Thanks for having us.
Sure.
Looking forward to our discussion.
All righty. Yeah, me too. Can you begin by telling us a little bit about yourselves and your organizations, starting, Michelle, with you?
Sure. So I'm Michelle Gilson. I'm one of the senior analysts at Canaccord Genuity, and my colleague Arlinda is another one of the senior analysts at our firm. We cover the biotechnology sector, and my more specific focus is on rare disease. So as equity research analysts, we make recommendations on the stocks of companies within the biotechnology and therapeutics sector. And so because of my coverage group and the focus on rare diseases, many of them have genetic drivers. So I end up spending time evaluating emerging gene technologies. So a bit about, I guess, our firm, Canaccord's a global mid-market investment bank. We have sort of, I guess, a full suite of services that go along with that, including sales and trading, equity capital markets, banking, and obviously research, which is what Arlinda and I do. We also have a wealth management arm, and we're traded actually on the Toronto Exchange. We have a really strong balance sheet with $650 million working capital. And our focus at Canaccord is really on growth sectors, biotech, and more broadly, healthcare, tech. We have space and sustainability. And so we like to stay ahead of, I guess, emerging growth sectors.
Mm-hmm. And Arlinda?
Great, thanks. Yeah, so I joined Canaccord about 2015 to cover oncology and rare disease. I work alongside Michelle and 3 other analysts at Canaccord. Our focus generally, as Michelle said, is on emerging growth companies globally. Michelle and I overlap a bit on the gene therapy space because we both cover rare disease. I think a little bit more so Michelle than me at this point. I have more oncology coverage, but I do cover CRISPR, and we should note that they've been an investment banking client in the last 12 months.
Arlinda, Michelle, what interested you both in the biotech industry as an industry to cover?
So I was actually really pretty young when I started on the sell side. I think I was 20, actually 21 probably, as an undergrad when I did an internship in biotech equity research. So I really fell in love with the space. During that internship, you learn something new every single day. It's very dynamic, and the people are really amazing. You have all of these creative, brilliant folks that are in the biotech industry working towards this mission to bring new therapeutics to patients. It's very inspiring work they do, and can change people's lives. So I feel really fortunate to be able to work with the companies that I cover, and even if we're more on the sidelines, I guess, of these innovations on the sell side research perspective.
Geulah, how and why did you transition from the lab to Wall Street, so to speak, going from a career in science to a career as an equity analyst?
Yeah, great question. So I guess just in terms of the intro and the explanation of the narrative. So as you mentioned in the intro, I've been at Chardan for about 3.5 years now. And before that, the background was about 12 years in academic science. And kind of the common thread in the academic life has been genetic engineering. So I did the PhD at Rockefeller, where I used in vivo lentiviral delivery to study gene function in skin regeneration and development with RNAi technology. Then I did the postdoc at Memorial Sloan Kettering Cancer Center, where I studied the pancreatic cancer models. And that's where I was first introduced to CRISPR technology and was able to use that. And so as a senior analyst at Chardan, I've been fortunate to be able to leverage that scientific background in my coverage in the genetic medicine space, so which includes the genome editing companies like Intellia, Editas, and Crispr Therapeutics, as well as some cell therapies, RNA names, and vector gene therapy names. And so kind of in terms of the trajectory, I knew that I liked science and wanted to continue to be close to really exciting scientific advances and read papers and talk to interesting scientists that were making these advances. And equity research seems a great way to be able to stick close to the really exciting applied science while also branching out in an area of something new, and also helping investors make good decisions with respect to their investment returns by leveraging my, again, scientific background. So I guess more broadly on Chardan, Chardan's a boutique investment bank focused on disruptive innovation, and there's a strong footprint in the biotech industry, and in particular genetic medicines, microbiome, RNA medicines, and some other areas of innovative biotech. And the model has really been to identify companies with the potential to generate 5x and 10x investment returns by creating value for patients and for society. And so it's been really exciting to see the gene editing space coming into alignment with that on multiple fronts.
Do you miss the bench at all?
I get asked that a lot, actually. And no, I actually find that I read more papers now, or at least glance through more papers now than I did as a postdoc, because, as a scientist, there's a lot of really exciting research that you're doing yourself, but it's also quite a bit more narrow. You're focused on your project. Maybe you talk to your colleagues about their projects. and you go to lectures, but it does end up being more of a narrow focus. Whereas on the equity research side, I find that, as I say, I have a 10,000-foot view of the biotech space and understanding the dynamics, what works, what doesn't, what communication is like, and how that's perceived. And I think it's been really interesting to learn all of those dynamics and follow along as the companies that I cover have progressed.
Mm. Now, all three of you devote some significant time to coverage of the genome editing space. There was a report released last week projecting the field would grow to more than 15 billion by 2027 from 3.7 billion last year, based on a 23% compound annual growth rate. Now, putting these numbers aside for a moment, how do you assess the state of genome editing right now?
I think that, the state of gene editing right now is it's a really exciting time. I think we're just really in the beginning stages. This proof of concept for in vivo gene editing in the clinic by Intellia that was reported, that was really groundbreaking. But I think, one of the things that's underappreciated is that this technology moved really quickly. Intellia IPO'd in 2016. And, now with the modularity that Crispr offers, they can continue to build off what they've done. But then, in terms of maturity and like where we are now, there's, challenges ahead to getting beyond just knockout, to getting outside the liver. We're, at the beginning stages of expanding this toolbox that, we have in terms of, CRISPR Cas9 systems and getting, beyond just, what you can do with Cas9. So, there's going to be more specificity, more modularity, more diverse capabilities in the next generation of tools, so to speak, and, some really great companies and smart people working on this. I think that as we have more tools coming into our gene editing toolbox, the capabilities of what we can do are going to expand to be more meaningful to patients.
I would echo that. I think it's a very, the data point that everybody's referring to, I think was very exciting for a lot of people, but, I guess just taking a step backwards, I remember, being a little undergrad working at Genentech, and we were working on gene therapy back then, right? And, we've certainly come a long way since then. The bubble boys clearly, that was not proven so successful, but, gene therapy and gene editing has kind of started in this new era more recently. When we first joined Canaccord, we had talked about this space as a potential to cover. And I think one of the pushbacks we got was like, well, this is just so early. And this was not even 5 or 6 years ago, right? So it's certainly come a long ways. And I think we're at the cusp of some very interesting discoveries and technological advances that is going to enable this to become not just a bench experiment, right, but a reality in terms of therapeutics.
Yeah, I'll just add quickly that, back in 2018, at Chardan, we put out this gene editing framework where, we're getting questions on how do you value gene editing companies, particularly because at that point, all the companies were preclinical. And so one of the core hypotheses was that gene editing approaches could be able to move quickly by being able to leverage the technological learnings on delivery from the AAV and the RNAi space, as well as the manufacturing and cell processing approaches for the engineered cell medicine space. And so taking that existing technology and then piggybacking the gene editing technology on top of that would enable the companies to move more quickly towards into the clinic and develop proof of concept by kind of reducing some of the unknowns around technology that have been a challenge in this space without having to kind of reinvent the wheel on that front. And we're kind of in the midst of that inflection point in the space right now, and it's been quite exciting. And as my colleagues have said, the space is continuing to move forward with enhanced capabilities with respect to novel editing enzymes that are being discovered, novel capabilities that are being engineered onto the existing enzymes, as well as novel delivery technologies that are being advanced to get into tissues beyond the liver and the eye, et cetera. And I'm sure we'll touch on all of this.
And just looking at it How mature is this technology? How ready is genome editing to deliver on years of promise, sometimes hype, about being able to deliver curative treatments for a range of diseases?
Well, I think, the key advantage and point that has inspired people about gene editing is that it is permanent genome modification. So the idea is, in theory, that, with a one-shot treatment, you can potentially, permanently modify the genome to improve the clinical profile of a particular therapy. So now there's additional complicating factors, and that will vary by different disease indications. For example, for cell-based therapies, the curative potential also relies on the fact that the cells need to engraft permanently rather than become depleted over time. And so that's something that's outside the specific elements of the gene editing technology and more on the cell processing and conditioning front. And there's been good progress on that within the hematology space for sickle cell disease and beta thalassemia, where we've seen durable cell therapy. And so there's no reason to think that the gene editing based approaches would see anything different. But with each type of indication, there's going to be its own questions regarding the permanence. For other indications, for example, if the edited cells have some kind of a competitive disadvantage, they might be lost over time, or if the editing event creates some kind of a protein that could be recognized as foreign by the immune system, they could be cleared that way. So those are kind of the types of areas where there could be limitations, but at the same time, everything that we've seen to date, including Intellia's data that they've reported in mice and non-human primates, point towards durable effects of the genome editing therapies.
Mm-hmm.
Well, let's talk a bit more about the Intellia study. Michelle, you brought it up a minute ago. This was published 2 weeks or thereabouts ago from the time we're recording this in the New England Journal of Medicine. Intellia, in partnership with Regeneron, publishing some of the first clinical data in a small clinical trial for a rare liver disease using CRISPR-Cas9, handful of patients, 6 patients, and by all accounts, pretty spectacular results. Michelle and Arlinda, would you like to just sort of flesh that out and just summarize for us, what stood out for you about that particular study?
Actually, maybe because I know Geulah actually covers Intellia, so it might be great if Geulah shares her thoughts first.
Sure.
Sure. So do you want a summary of the study itself, or
program? Or yes, I think briefly, what did they do, and why did it cause so much excitement? It seems to have gone down. It seems to be a real landmark in the field.
Mm-hmm. Yeah, absolutely. So the program is in transthyretin amyloidosis, or as you mentioned, a rare hereditary liver disease, and the approach that that the program takes is using lipid nanoparticles to deliver mRNAs encoding the CRISPR components intravenously, and then they get taken up by the liver, expressed in the liver, and then edit the target of the CRISPR in hepatocytes. And so in preclinical models, this has been shown to reduce the transthyretin protein, which is the thing that causes the disease phenotype in these patients. And so they've shown that they have been able to reduce the circulating levels of the target protein, as well as improve clinical disease phenotypes in the animal model, the mouse model, I should say. And they've also shown in non-human primates beforehand that they can get similarly high levels of editing and durably over the course of a year. So I should say that this target is one that's been well covered in the RNAi space. Alnylam's Onpattro targets the same gene, and but the difference is that it's an RNAi approach, so it's not a permanent one-and-done solution, but it's also delivered in a lipid nanoparticle formulation intravenously. But in that setting, it's not a one-and-done. It has to be delivered, every few weeks. But the target has been validated from that standpoint. And Alnylam has the next-gen version that allows for less frequent dosing, but again, going after the same target. So now Intellia has shown, number one, that they can get reductions of this transthyretin protein to similar levels that Alnylam has shown with Onpattro at levels that are associated with clinically meaningful effects in people. And secondly, the thing that I thought was important here is that basically we see good translation from the preclinical setting into humans. And, within the biotech space, that's often an area where, you see great data in mice, and then it just doesn't happen in the human setting. I come from an oncology background. It's very common to cure cancer in mice, and then it doesn't happen in humans for various reasons, including heterogeneity of cancer, unknown factors, et cetera. So the fact that the data translated well from mice to non-human primates and humans now, even with respect to the extent of biomarker reduction that they saw that could be modeled, was a kind of a strong signal that provides some de-risking to the LNP CRISPR platform and also makes future preclinical data more meaningful.
Michelle, go ahead.
Oh, I was going to say, I couldn't agree more. From my perspective, I really think that this read through from non-human primates to humans, just we can't discount this. And I think, it also showed us that LNPs are probably going to be a powerful tool going forward to deliver these gene editing components. They translate very well from non-human primates to humans. We've seen that not only with, siRNA and mRNA for Cas9 and guide RNAs is, the mRNA vaccines as well. So I think that, that translated really well, but also, it showed that you can optimize the guides, for human cells to increase the potency. They really had high efficiency here for editing. It blew me away. And on the safety side, they didn't see an immune response to Cas9. This has been a theoretical risk that's, persisted for a while. And it showed that, that transient expression delivering Cas9 mRNA, maybe mitigates this risk. We'll see, with more patients, and then, long-term risks off targets, things like that. We're going to see how that progresses in the future as well. So, I think that this was really, it was a landmark study.
Yeah. Yeah. Arlinda, anything you'd like to add to that? I think we've covered a lot of it.
I think they've covered almost all of it, but I would agree with all of those points. This provided a great, it was a landmark study because I think it provided proof of concept for everybody else that this indeed was possible and that we got the validations that we wanted, and it de-risked a lot of the theoretical risks that we were all worried about. So I think it's, I think for all of the programs, it becomes validating.
Yeah. Yeah. One of the interesting aspects of the immediate aftermath of the publication, it was not just the spike in share price that Intellia received, but it just lifted all the other boats along with it. Were you expecting that? It seemed to speak to something that I think you've all touched on, which is this wasn't just about one company or one disease, but about a much broader sense that the delivery system is safe. And there's many more diseases that now are amenable to this strategy. Would love to hear your thoughts about what happened over the last 2 weeks.
Well, from the CRISPR perspective, I guess, since we cover CRISPR and not Intellia or Editas, I would say that we did get a lot of inbound questions about what this means for CRISPR and what programs specifically CRISPR has in the clinic that could address this. And, you know, they don't have anything in the clinic about this, right? And so, you know, we got questions about what this means for them. And it's like, well, I mean, I think for not CRISPR specifically, but everybody in general, this opens a whole new set of diseases that are amenable to treatment with this new technology, right? And whether it's Intellia doing it or CRISPR or somebody else, this is now,
of course, CRISPR Therapeutics' big success in sickle cell has been an ex vivo approach. Is that correct?
Right. Absolutely correct.
Yeah.
Absolutely correct.
Okay.
So,
yeah. But it was fascinating to see. I mean, even non-CRISPR company. I mean, Beam is sort of a CRISPR company, but it's a different technology. They had a good run. I guess a lot of these gains have been given back. But how do you this choppiness, this sort of roller coaster? Is this just part of the game, or do you how does that impact your sort of? Don't you have to sort of set prices and give predictions as part of your job? Must make life pretty difficult.
I mean, I think in biotech, there's a little bit more, or quite a little bit more, hand waving in terms of valuations or potential peak valuations for these kinds of technologies, right? And certainly we have people put multiples on depending on what kinds of validation there is. And we have not updated our model for this, but certainly I don't think it would be unreasonable to see people increasing multiples because of validating, or increasing probabilities of success because of validating technologies from competitor companies for similar technologies.
Okay. Geulah,
maybe I'll let, sorry, I was going to say maybe Michelle or Geulah would love to weigh in on how they think about that.
It just makes your job extra fun, I'm sure, to see these perturbations.
Yeah. No, absolutely. I mean, I think there's areas where there's specific read across to companies that are using similar technology. For example, you mentioned Beam. There is a more direct read across. I don't cover Beam, but in general, they're also using lipid nanoparticle technology to deliver CRISPR components into the liver. So them and Verve as well, there's more direct read across on the science. Again, I can't speak to valuations, they're not covered companies, but scientifically speaking, it provides, as I mentioned, broader validation for the lipid nanoparticle based delivery of CRISPR components to the liver in vivo. So companies directly working on that have that additional layer of de-risking of the.
Just wanted to follow up on that. So the lipid nanoparticle aspect of the story, is that primarily of interest to companies and therapies that are targeted to the liver, or will that have relevance for other tissues and other parts of the body as well?
Great question. So right now, that's largely focused on the liver, and that's the space where there's been the greatest progress on getting things to the liver. As I mentioned, there's an approved therapy. Moderna has put RNA therapeutics, I should say, that have been driven systemically and expressed in the liver, et cetera. But there is a lot of interest in moving beyond the liver, looking ahead. For example, Intellia actually has reported some very early preclinical data using lipid nanoparticles to deliver CRISPR components to the bone marrow. So lipid nanoparticles, the way that they get taken up by the liver by their biophysical properties and tuning the properties of the lipid nanoparticles can skew them towards uptake by different tissue types. The details behind that, I think we don't have full visibility on, but that seems to be an active area of research. I think Beam has also announced the acquisition of Guide Therapeutics, which may also enable delivery to other organ systems. So, I thought that's an area that we're likely to hear a lot more about with regards to non-viral delivery to various organ systems. Although for now, I would say delivery to areas outside of the liver is dominated by viral-based approaches.
I'd like to ask all of you, in the last 6 months, we've had 2 landmark clinical studies, both in the New England Journal. We've had the Crispr Therapeutics sickle cell study published at the end of last year, and now, of course, the Intellia study. Are there other clinical milestones in terms of peer review publications that are on your radar? Are you sort of, have you got your calendar circled for certain key papers that maybe, I'm not asking you to sort of give us notice, but what are some of the big trials that you think will provide other potential milestone moments for this field as a whole?
So I can start just briefly. So a couple of things in terms of, I'm not sure about papers, but in terms of data readouts, ones that are important for example, for the in vivo editing space, Editas is in the clinic with in vivo gene therapy. It's not systemic, it's delivered locally to the eye, and it uses an AAV vector. And so they've announced plans to report data this year, so we can see some initial data on that. So that would be data with an in vivo AAV-based CRISPR approach. And then also outside of the CRISPR space, Logic Biotherapeutics, which we also cover, have had investment banking relationships with in the past. They also have viral nuclease-free technology that they're in the clinic with for a rare disease called methylmalonic acidemia, and they've announced plans to report initial data on that in pediatric patients by the end of the year. So some potential for data on viral gene editing approaches in vivo this year, and additional durability data on some of the cell-based approaches, I would say, is on the horizon. For example, for Crispr Therapeutics, they reported initial data in their CD19-focused CAR T program, but we're expecting to see durability data on that, so longer-term follow-up, as well as some initial data in their CAR T programs targeting other targets in the space.
Excellent. Michelle and Arlinda, anything else you wanted to add to that?
I would just say that for CRISPR, I think we're not really expecting any in vivo edited data flow, but in terms of the ex vivo edited things, we are expecting, I think, to your point with the New England Journal paper of their lead compound CTX001, that should be, wrapping up enrollment fairly soon. So we might get something by year end, but more likely next year. And then for their 3 cell CAR T therapies, CTX 110, 120, and 130, targeting CD19, BCMA, and CD70. I think those are all should be having at least initial data or data updates all this year as well.
Okay, great. You're watching Gen Edge Live. We're talking to Michelle Gilson and Arlinda Lee from Canaccord Genuity and Geulah Livshits from Chardan. And Alex, we welcome your questions. Send them our way and use the Q&A box. Alex, over to you.
Okay, sure. And there are a couple of questions that we have that I'll read through and bring to our guests. One, this is from Matthew to all panelists. What does Intellia's data mean for the upcoming Verve trial?
I think I kind of touched on that maybe in one of my earlier comments, but yeah, I mean, I think that Intellia's data has broader read across for the translatability of the LNP CRISPR space in terms of in vivo editing. Again, as I said, obviously every target is going to be different and have its own threshold of knockdown that's needed to see a clinical effect, but it increases broadly the confidence in the translatability of LNP CRISPR data from non-human primate to human.
There were some points earlier in our discussion that I wanted to get a little bit of elaboration on. One was that genome editing had focused on disorders of the liver, but is more likely preparing to broaden into other indications, other parts of the body. Which other indications and disorders are we most likely to see genome editing branch to first, and why?
So I guess to start, there are a few things we should probably point out about the Intellia approach. They chose a hepatocyte target, right? LNPs go to hepatocytes. So there was some cell specificity as well as organ specificity there. They chose also an established target. I think Geulah touched on this. We've seen the benefit that's associated with TTR knockdown in the siRNA and ASO data. And so, they were really smart. They chose these kind of low-hanging fruit targets, I guess, to validate the platform. I'm not minimizing in any way the accomplishment here, but, a lot of other disease targets, they're going to be more challenging. And so moving beyond the liver is really going to be mediated by the next generation of delivery technologies. I think, Geulah mentioned the Editas data. That's an AAV-based approach. And so, there have been efforts to, I guess, deliver AAV beyond the liver as well. And so where AAV, if that approach is successful, we would definitely see probably the expansion of that approach. We've seen AAV targeted to muscle, we've seen AAV targeted to the CNS. And so I wouldn't be surprised to see, those kinds of approaches next. And then, on the LNP side, there are companies that are developing the next generation of LNPs. We've seen those read through pretty well in terms of the liver targeting, but there's a company called Verve Therapeutics. They're working on a next generation of LNPs that go beyond the liver, and some more organ specific targeting to organs like the lung and the spleen. And so I imagine we'll see the emergence of, I guess, the next generation of LNPs and other delivery technologies like exosomes and things like that that are really being worked on to get some of these tools beyond the liver.
No, I totally agree. I think, as I said before, I think gene editing is really piggybacking on the delivery technologies that have been advanced for other approaches. So again, as Michelle said, we've seen AAV gene therapy in muscle, CNS. We've seen different approaches attempting to go into the lung with lipid nanoparticles at Moderna, which we also cover out the collaboration with Vertex that includes potential gene editing in the lung. So clearly there's interest there. And yeah, more broadly, anywhere where people are thinking about delivering AAVs and lipid nanoparticles, we can see as areas of interest. Again, as I mentioned earlier, also the bone marrow is an area of interest. The Gates Foundation has given out a number of grants to focus on developing in vivo therapies for sickle cell disease because those are operationally easier to execute. Because it doesn't involve a cell transplantation, et cetera. So we could see additional interest there.
Mm-hmm. And have we come to a point where we know that certain delivery methods work better for certain organs versus others? LNPs for one or AAVs for other? Or is it still kind of a loose or uncertain field?
I think it's, at a high level, it's still evolving. I think, LNPs, we now have data that suggests that LNPs work in liver. The AAV delivery also does work in the liver, but, with CRISPR, one of the advantages of LNP technology is that you deliver the editing enzyme. It's there transiently, does the edit, and then it's cleared. You don't necessarily want to have a long-term expression of a nuclease in a tissue that it's not fully differentiated and could still divide. I think that we've said that there are advantages to having this more transient expression in the liver that you get with LNPs. That's not to say that you can't use AAV approaches to deliver, and there could be scenarios where it might make more sense to deliver something as an AAV vector, but certainly for other tissues where LNPs haven't yet shown successful delivery, AAV and potentially AAV seems to be the most mature technology, though there could be other viral approaches that in theory could be taken to deliver nuclease editing components. For example, in theory, one could use HSV vectors, or Sana has developed its fusosome technology, which could in principle also be used to deliver things in vivo. We don't cover Sana, or Sana, but just in principle, that can be used as well on the in vivo side for targeted delivery.
We've seen combinations too, right? There's approaches to deliver the Cas9 mRNA by LNP and using AAV to deliver the single-stranded DNA replacement strand for gene insertion. So, I think that LNPs are really going to have some staying power. What we're seeing as the field evolves, right, is that these systems are becoming more complex, not less complex. And there's probably more components that are going to be needed to deliver into one cell. And, I think LNPs are really, emerging as a way to do that. There may be others too that emerge, but I think that, going forward, the trend seems like it's towards more complex systems.
Mm-hmm. Yeah. I just forgot to mention that I also wanted to add in on LNPs in the liver. One of the other potential advantages that hasn't thus far been needed in the Intellia program, but could end up being useful later on, is that they can be redosed. As of now, redosing of AAVs has been challenging in the space because you develop an immune response against the vectors. So in theory, CRISPR therapy is a one-time treatment, but if a patient doesn't hit the optimal editing level on the first run, in theory, you can tune up the editing with successive rounds of administration of an LNP CRISPR, and that's something that's more challenging to do with an AAV-based approach. So again, this is not something that's been implemented yet in the clinic, but it's been shown in preclinical models and something that could be used down the line.
When you mentioned on the LNPs, to what extent might they go toward addressing some of the issues of safety that have taken place at larger doses involving AAVs, such as in the gene therapy space?
So, I mean, I would say thus far the data that we've seen from Intellia in terms of safety has been fairly clean. I think, the challenge is that, again, as of now, the LNPs have been largely limited to the liver, whereas the AAVs that are in wide use right now are not just focused on the liver. There's often systemic dosing where it's intended to be expressed in other tissues, including the central nervous system and the muscle. Again, those are areas that are not yet unlocked for the LNP system.
Mm. As with many other segments of biotech, genome editing has seen several companies go public over recent years, but especially in the past year plus. I mentioned earlier Verve Therapeutics. They generated more than $300 million in gross proceeds from their IPO in June. Caribou Biosciences just filed for an IPO last week. Their amount is yet to be determined, while Graphite Buyer began trading late last month, and they raised just about $274 million in gross proceeds. What's driving all this recent IPO activity, especially in the past year or so?
I don't think it's just gene editing that's been fairly hot. It's been a pretty strong biotech market all around. I think there's a lot of appetite on both the private and the public side for innovative new technologies to address these diseases, and particularly when you see clinical successes of companies or some kind of validation. I think people are more inclined to invest in maybe earlier programs or tweaks on a system that they view as being validated, right? So I don't think it's particular to gene therapy, but certainly I think the Intellia data from a couple of weeks ago should help fuel additional interest in gene editing going forward.
And I guess what effect, if any, does this IPO activity have on down the chain, so to speak, a new company formation and financings? Do these companies going public at later phases influence and stoke more company creation earlier on?
I think so, but I think it also accelerates what companies that are already, I guess, have technologies they're developing in this space, how quickly they can move. Some of these de-risking factors, like what we mentioned with delivery, for example, I think have really prompted companies to accelerate their gene editing pipelines, and investors to also be willing to invest earlier in them. So, I think that as these advances, as gene editing starts to mature, you're going to see more, more tools coming down the pipeline here, both on the private and public side. I wouldn't be surprised to see some consolidation as well for some of these sort of one-off tools, like some novel nucleases, and in different, I guess, approaches to delivering replacement strands, things like that. And then, for some of these bigger, I think, platform technologies that are sort of the next step edit advances, I guess, pun intended. I think that you'll start to see those companies becoming pretty, big platform companies, which is, what we saw with the first generation, with Intellia, with Crispr, with Editas, the formation of those companies.
Well, we see more platform companies as opposed to straight out therapeutics developers on this, or at least as many. Is this more platform driven than drug driven?
I think for now, but that's how early stage companies start, right? With platform technologies. And then you find the best fit for your technology in the way that can be most impactful, I guess, down the road for human health and disease.
On Gen Edge Live, we're on with Geulah Livshits of Chardan and Michelle Gilson and Arlinda Lee, both of Canaccord Genuity, 3 analysts. We're talking genome editing, and we do have an audience question. You're welcome, by the way, to submit your questions in the Q&A box of Zoom. We do have one from Joe, who asks, are there any specific private companies you like from the therapeutics, genetics, or bioinformatics space that are not within your public coverage universe?
I'll mention 3 here. There's a company called Genome that has a really powerful platform. It's driven by their metagenomics-based discovery engine. They've discovered novel editing systems with new properties and new capabilities. They have novel CRISPR-based nucleases, expanding what you can do with nuclease-based editing. So they can target different sequences that aren't limited by the Cas9 PAM sites, and they can make staggered or blunt cuts. These CRISPR-associated systems, they can really engineer them in a modular way. So overall, new properties, equals new capabilities for these CRISPR-associated systems. It allows them to, pick a system that fits what they're trying to do, that fits the disease that they're trying to modify. They also have discovered new base editors for correction and some transposon-based systems for targeted integration. So I think that that's, really cool, very big ideas there. There's a company called Recode Therapeutics I mentioned before. They have novel LNPs. Their LNPs are 5-component LNPs instead of 4, which is what was used for kind of the first generation. They have some better organ targeting and cell selectivity. If we want to get beyond hepatocytes, I think that you need some new delivery systems, as we've said. LNPs, they'll have some staying power. Another really cool aspect to their platform is they can encapsulate more complex cargo, including RNPs, not just nucleic acid cargo. So our systems are getting more complex. We're going to need to be able to deliver all of these pieces to the right cells. I think that there's another company called Coro, another really important technology. They are focused on RNA editing. We've mentioned all these problems with delivery, complexity of cargo. Moving to RNA offers a lot of modularity in how you affect expression. They're co-opting a natural enzymatic mechanism. It's ADAR-based, and this is really elegant. They can do base editing to fix genes at the mRNA or the translation level, where you can affect essentially an A to G edit, but they can also modify expression in other ways since they target RNA, non-coding regions, modifying protein-protein interactions, introducing protected mutations. So it's a lot of the benefits of gene editing, but also the benefits of targeting RNA, in terms of safety, it's transient editing, modularity. They're using oligonucleotides here. We know that we can deliver oligos, right? And these synthetic oligos are designed to co-opt the endogenous ADAR, what we have in our cells and our bodies already. We have these ADAR proteins in our cells, so you just have to direct them to where you need to make the edit. So I think that those are some three very cool companies that are on the private side.
Great. Kevin, how are you?
Thanks, Alex. We've got a little bit of time left. I wanted to just quickly circle back and sort of touch on a couple of companies that that Alex mentioned a few minutes ago. Just get your quick take on their prospects. You may not be covering them yet, but Verve Therapeutics is a very interesting company because unlike pretty much every other company we've been talking about, they're not tackling a rare disease. They're tackling a much more common disease. I'd love to get your take briefly on what you think the prospects are for gene editing for cardiovascular disease. Geulah, shall I start with you?
Sure. So, again, I don't cover them, so I can't comment on valuation, but, in general, from a scientific standpoint, for every genome editing approach, each is going to have its own risk-benefit assessment for each individual indication. Again, with Verve's program, they're going after, as Intellia was, something that is a known and validated target within the RNAi space. And so there's additional parallels there. But again, the specific risk benefit for each indication is really going to depend on the sentiments of doctors and patients and how they view the advantage of a one-time administration with the balance of potential unknowns with regard to a permanent genome modification. And there's clearly interest and appetite within the space based on enthusiasm. But those are all things that are going to be considered as the program moves forward.
Great. I also wanted to ask Michelle, and I'll put this one to you first of all. Alex mentioned Graphite Buyer. This is a relatively new company, co-founded by Matt Porteous at Stanford, and they're going after sickle cell disease, as are many gene editing companies, including Crispr Therapeutics, of course. And it's great to see the interest in sickle cell, which has been such a neglected, appallingly neglected disease from a sort of therapeutic standpoint. So I think that's wonderful. But I'm curious whether you feel this area is getting a little bit crowded and whether that could potentially lead to a little bit of sort of winners and losers and head-to-head competition just at the infancy of this field. Is it getting too crowded, or do you welcome all these different approaches for one disease?
I would say that sickle cell, I think, remains a pretty large unmet need. And I think the approaches that we've seen so far all kind of make sense, right? That you need that and recognize the fact that you do need to address this pretty severe disease. I know that people think of things as a zero-sum game, but I think, frankly, the more drugs that are available that are effective, the better for the patients. So yeah. I think that companies should continue to innovate. And if sickle cell is one where that's what they choose to go after, I mean, it makes scientific sense, right? Yeah. And if they need, and actually, from an analyst perspective, you have a lot of other therapies that you have, like phase 1, phase 2, phase 3 data for. And so it actually makes our life a little bit easier, because then we're trying to benchmark against things that have come before.
I would also love to get your quick take about the news today about the launch of Prime Medicine. Well, the company got off the ground 18 months ago when David Liu and Andrew Anzalone published their brilliant Prime editing technology in Nature at the end of 2019. We've been waiting for the official launch of the company. It happened this week, today, with a $315 million Series A raise. So for a long time, gene editing was, in terms of publicly traded companies, was just 3 companies. And obviously Prime isn't public yet, but one imagines it may not be too long. What are your thoughts about the flexibility and the potential of the Prime editing platform? Geulah, I'll start with you and then see if anyone else wants to join in.
Sure. So I remember when the initial paper came out, it was super interesting. I remember actually going to write up and write a note to investors about, what it is and what it isn't and the potential there. And so, it's a really interesting expansion of the genome editing toolbox, and in that it's a little bit different. It's like base editing in that you hook something else onto a CRISPR enzyme and have it add its own functionality, but it's different in that base editing can only make certain conversions and prime editing can make a broader list of corrections, so it can correct a variety of gene mutations, as well as do small insertions and deletions, et cetera. So it's really, an exciting technology, and in terms of the platform, again, I think coming out of the initial publication, there was a number of open questions, as there usually are when a new technology is developed, with regards to efficiency. I think, again, as with the initial CRISPR technology, when it was developed, the efficiency was low, and since then, companies have, and academic groups have worked to optimize the efficiency of CRISPR editing, both on optimizing the nuclease part and on guide design and algorithms and things like that. So it would be very interesting to see how the platform has advanced in terms of efficiency. We also have some open questions on delivery and things like that. It would be a large enzyme, might be difficult to fit it into AAV, but with lipid nanoparticles in vivo, in theory, that could be possible. And then on off-target editing, where the editing the characterization of off-target editing with each different type of technology might be a little bit different, but overall, very powerful technology that seems very interesting and a great addition to the toolbox. So we'll definitely look forward to hearing more on that as it develops.
We sure do. Michelle, did you want to add something?
Oh, yeah, I would say that I think that it's also really exciting, as Geulah mentioned, you maintain the targeting of CRISPR-Cas9 systems, and they kind of piggyback as an extension to the guide RNA, an RNA template for reverse transcriptase, which is fused to the Cas9 nickase, so they're not creating double-stranded breaks. And so, using the RNA template as the primer for reverse transcriptase, I think that is great, because reverse transcriptase has been like the enemy to humans, right? And now we're co-opting it. And so, I think that, what they've shown is that you can make these insertions. They're not, another thing that I think is not, hasn't been mentioned is that, you're not using HDR. And so you maybe can expand the types of cells that you target beyond because they use, I think from my memory, they use mismatch repair. So you can expand, I guess, the stage of the cells that you're targeting, and to maybe cells that don't have the HDR machinery. And so I think that, improving the efficiency, as Geulah said, and predicting the off targets are going to be important. Delivery, and then also, expanding the PAM site. You can only make the edit near where the PAM site is. So I think the efforts to increase PAM flexibility is going to be important as well.
Yeah, a very exciting space, and yes, the toolbox just gets more and more crowded, which is really exciting. We're right at the top of the hour. We had pages and pages of other questions. We wanted to get into pricing and delivery and many other topics, but I think we'll have to leave it there. Alex, I'm going to pass it back to you.
Sure. Thanks, Kevin. And just like that, that's all the time we have today. Thank you, Michelle Gilson and Arlinda Lee from Canaccord Genuity. Thank you, Geulah Livshits from Chardan, for your fascinating insights today. And to all of you watching and listening, please check out our other new Zoom series, Close to the Edge, featuring interviews with outstanding biotech chief executives, including Ted Love of Global Blood Therapeutics, John Evans, and Daphne Zohar.
Gen Edge Live is produced by Jamie Cohen and Danny Buda. For Alex Philippidis, I'm Kevin Davis. Thanks so much for watching. Goodbye for now.